Class 11, Chapter 8 Cell, the Unit of Life. When you look around, you see both living and non living things. You must have wondered and asked yourself, what is it that makes an organism living, or what is it that an inanimate thing does not have, which a living thing has? The answer to this is the presence of the basic unit of life, the cell, in all living organisms. All organisms are composed of cells. Some are composed of a single cell and are called unicellular organism, while others, like us, composed of many cells are called multicellular organism. 8.1 What is a cell? Unicellular organisms are capable of number 1 independent existence and number 2 performing the essential functions of life. Anything less than a complete structure of a cell does not ensure independent living. Hence, cell is the fundamental structural and functional unit of all living organisms. Antony von Leeuwenhoek first saw and described a live cell. Robert Brown later discovered the nucleus. The invention of the microscope and its improvement leading to the electron microscope revealed all the structural details of the cell. 8.2 Cell Theory. In 1838, Matthias Schleiden, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells which form the tissues of the plant. At about the same time, Theodor Schwann, 1839, a German zoologist, studied different types of animal cells and reported that cells had a thin outer layer which is today known as the plasma membrane. He also concluded, based on his studies on plant tissues, that the presence of cell wall is a unique character of the plant cells. On the basis of this, Schwann proposed the hypothesis that the bodies of animals and plants are composed of cells and products of cells. Schleiden and Schwann together formulated the cell theory. This theory, however, did not explain as to how new cells were formed. Rudolf Virchow, 1855, first explained that cells divided and new cells are formed from pre-existing cells, omnis cellula e cellula. He modified the hypothesis of Schleiden and Schwann to give the cell theory a final shape. Cell theory as understood today is number 1 all living organisms are composed of cells and products of cells, and number 2 all cells arise from pre-existing cells. 8.3 An overview of cell. You have earlier observed cells in an onion peel and or human cheek cells under the microscope. Let us recollect their structure. The onion cell, which is a typical plant cell, has a distinct cell wall as its outer boundary, and just within it is the cell membrane. The cells of the human cheek have an outer membrane as the delimiting structure of the cell. Inside each cell is a dense membrane bound structure called nucleus. This nucleus contains the chromosomes, which in turn contain the genetic material, also known as the DNA. Cells that have membrane-bound nuclei are called eukaryotic, whereas cells that lack a membrane-bound nucleus are prokaryotic. In both prokaryotic and eukaryotic cells, a semi-fluid matrix called cytoplasm occupies the volume of the cell. The cytoplasm is the main arena of cellular activities in both the plant and animal cells. Various chemical reactions occur in it to keep the cell in the living state. Besides the nucleus, the eukaryotic cells have other membrane-bound distinct structures called organelles like the endoplasmic reticulum ER, the Golgi complex, lysosomes, mitochondria, microbodies and vacuoles. The prokaryotic cells lack such membrane-bound organelles. Ribosomes are non-membrane bound organelles found in all cells, that is both eukaryotic as well as prokaryotic. Within the cell, ribosomes are found not only in the cytoplasm but also within the two organelles which are chloroplast in plants and mitochondria and on a rough endoplasmic reticulum ER. Animal cells contain another non-membrane bound organelle called centrosome which helps in cell division. Cells differ greatly in size, shape, and activities, figure 8.1. For example, mycoplasmas, the smallest cells, are only 0.3 micrometer in length, while bacteria could be 3 to 5 micrometer. The largest isolated single cell is the egg of an ostrich. Among multicellular organisms, human red blood cells are about 7.0 micrometer in diameter. Nerve cells are some of the longest cells. Cells also vary greatly in their shape. They may be disc-like, polygonal, columnar, cuboid, thread-like, or even irregular. The shape of the cell may vary with the function they perform. 8.4 Prokaryotic cells. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, and PPLO pleuropneumonia-like organisms. They are generally smaller and multiply more rapidly than the eukaryotic cells, figure 8.2. They may vary greatly in shape and size. The four basic shapes of bacteria are bacillus, which is rod-like, coccus, which is spherical, vibrio, which is comma-shaped, and spirillum, which is spiral shaped. The organization of the prokaryotic cell is fundamentally similar even though prokaryotes exhibit a wide variety of shapes and functions. All prokaryotes have a cell wall surrounding the cell membrane except in mycoplasma. The semi-fluid matrix filling the cell is the cytoplasm. There is no well-defined nucleus. The genetic material is basically naked, not enveloped by a nuclear membrane. In addition to the genomic DNA, which is the single chromosome or circular DNA, many bacteria have small circular DNA outside the genomic DNA. These smaller DNA are called plasmids. The plasmid DNA confers certain unique phenotypic characters to such bacteria. One such character is resistant to antibiotics. In higher classes, you will learn that this plasmid DNA is used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryotes. No organelles like the ones in eukaryotes are found in prokaryotic cells except for ribosomes. Prokaryotes have something unique in the form of inclusions, a specialized differentiated form of cell membrane called mesosome is the characteristics of prokaryotes. They are essentially infoldings of cell membrane. 8.4.1 Cell envelope and its modification. Most prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope. The cell envelope consists of a tightly bound three-layered structure, that is the outermost glycocalyx, followed by the cell wall and then the plasma membrane. Although each layer of the envelope performs a distinct function, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of differences in the cell envelopes and the manner in which they respond to the staining procedure developed by Gram, with those that take up the Gram stain are Gram positive and the others that do not are called Gram negative bacteria. Glycocalyx differs in composition and thickness among different bacteria. It could be a loose sheath called the slime layer in some, while in others it may be thick and tough called the capsule. The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing. The plasma membrane is selectively permeable in nature and interacts with the outside world. This membrane is similar structurally to that of the eukaryotes. A special membranous structure is the mesosome, which is formed by the extension of plasma membrane into the cell. These extensions are in the form of vesicles, tubules, and lamellae. They help in cell wall formation, DNA replication, and distribution of daughter cells. They also help in respiration secretion process to increase the surface area of plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extensions into the cytoplasm called chromatophores which contain pigments. Bacterial cells may be motile or non-motile. If motile, they have thin filamentous extensions from their cell wall called flagella. Bacteria show a range in the number and arrangement of flagella. Bacterial flagellum is composed of three parts: filament, hook, and basal body. The filament is the longest portion and extends from the cell surface to the outside. Besides flagella, pili and fimbriae are also surface structures of the bacteria but do not play a role in motility. The pili are elongated tubular structures made of a special protein. The fimbriae are small bristle-like fibers sprouting out of the cell. In some bacteria, they are known to help attach the bacteria to rocks in streams and also to the host tissue. 8.4.2 Ribosomes and inclusion bodies. In prokaryotes, ribosomes are associated with the plasma membrane of the cell. They are about 15 nanometer by 20 nanometer in size and are made of two subunits, 50S and 30S units, which when present together form 70S prokaryotic ribosome. Ribosomes are the site of protein synthesis. Several ribosomes may attach to a single mRNA to form a chain called polyribosomes or polysome. The ribosome of a polysome translate the mRNA into proteins. Inclusion bodies. Reserved material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. These are not bound by any membrane system and lie free in the cytoplasm. Example: phosphate granules, synophycean granules, and glycogen granules. Gas vacuoles are found in blue, green, and purple and green photosynthetic bacteria. 8.5 Eukaryotic cells. The eukaryotes includes all the protist, plants, animals, and fungi. In eukaryotic cells, there is an extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. Eukaryotic cells possess an organized nucleus with a nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeletal structures. Their genetic material is organized into chromosomes. All eukaryotic cells are not identical. Plant and animal cells are different, as the former, which is the plant, possess cell walls, plastids, and a large central vacuole, which are absent in the latter, which is the animal cells. On the other hand, animals have centrioles, which are absent in almost all plant cells, figure 8.3. Let us now look at individual cell organelles to understand their structure and functions. 8.5.1 Cell membrane. The detailed structure of the membrane was studied only after the advent of the electron microscopes in 1950s. Meanwhile, chemical studies on the cell membrane, especially in human red blood cells (RBCs), enabled the scientists to deduce the possible structure of plasma membrane. These studies showed that the cell membrane is mainly composed of lipids and proteins. The major lipids are phospholipids and are arranged in a bilayer. Also, the lipids are arranged within the membrane with the polar head towards the outer sides and the hydrophobic tails towards the inner side. This ensures that the nonpolar tail of saturated hydrocarbon is protected from the aqueous environment, figure 8.4. In addition to phospholipids, membrane also contains cholesterol. Later, biochemical investigation clearly revealed that the cell membranes also possess proteins and carbohydrates. The ratio of protein and lipid varies considerably in different cell types. In human beings, the membrane of the erythrocytes has approximately 52% protein and 40% lipids. Depending on the ease of extraction, membrane proteins can be classified as integral and peripheral. Peripheral proteins lie on the surface of membrane, while the integral proteins are partially or totally buried in the membrane. An improved model of the structure of cell membrane was proposed by Singer and Nichols, 1972, widely accepted as fluid mosaic model, figure 8.4. According to this, the quasi-fluid nature of lipid enables lateral movement of proteins within the overall bilayers. This ability to move within the membrane is measured as its fluidity. The fluid nature of the membrane is also important from the point of view of functions like cell growth, formation of intercellular junctions, secretion, endocytosis, cell division, etc. One of the most important functions of the plasma membrane is the transport of the molecules across it. The membrane is selectively permeable to some molecules present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called the passive transport. Neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient, that is, from high concentration to the lower. Water may also move across this membrane from higher to lower concentration. Movement of water by diffusion is called osmosis. As the polar molecules cannot pass through the non-polar lipid bilayer, they require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient, that is, from lower to the higher concentration. Such a transport is an energy dependent process in which ATP is utilized and is called active transport. Example, sodium plus and potassium plus sodium potassium pump. 8.5.2 Cell wall. As you may recall, a non-living rigid structure called the cell wall forms an outer covering of the plasma membrane of fungi and plants. Cell wall not only gives shape to the cell and protects the cell from mechanical damage and infection, it also helps in cell-to-cell interaction and provides barrier to undesirable macromolecules. Algae have cell wall made up of cellulose, galactans, mannans and minerals like calcium carbonate, while in other plants it consists of cellulose, hemicellulose, pectins and proteins. The cell wall of a young plant cell, the primary wall, is capable of growth which gradually diminishes as the cell matures, and the secondary wall is formed on the inner towards membrane side of the cell. The middle lamella is a layer mainly of calcium pectate which holds or glues the different neighboring cells together. The cell wall and middle lamella may be traversed by plasmodesmata which connect the cytoplasm of neighboring cells.